which we're quite interested in. But that can be quite complicated because you can actually get thin mice for a number of reasons, including effects on their fur and on their skin and their ability to burn energy. Another phenotype that might be quite interesting from the mouse point of view is resistance to diet-induced obesity. And again, a number of genes are now emerging that allow mice to retain a normal weight despite being fed a high-fat diet. I'd certainly like to have whatever this mouse has. And so what we've been interested in doing is trying to see if we could find a group of individuals who would be, if you like, resistant to a high-fat diet, who would be thin but well. And of course, finding these individuals is quite difficult because they don't necessarily come to medical attention. So one of the things we're very lucky about in the UK, of course, is our National Health Service. And with that, we have the clinical research networks, which allow us to recruit from general practice and primary care. And really, through a tremendous effort from my team led by Rebecca Bounds, we've been able to recruit, initially approach 25,000 thin people with a BMI less than 18, with an aim of recruiting a cohort of 1,500 thin people who are well with no eating disorders or other medical complications. And we're just now undertaking a comprehensive genetic study of these thin people so we can find common and rare genetic variants and ultimately characterize the phenotypes. And really just in the last couple of slides, our plan really is to try and take forward the insights about disease mechanisms coming from severe obesity and now also from thinness, and understand how they impact individuals at a physiological level by carefully studying the phenotypes involved. As a step, and I think again an invitation to collaborate, is we're building new clinical research facilities in metabolism in Cambridge, supported by the Wellcome Trust, which will include a new metabolic clinical research facility dedicated to these studies. But also, really importantly, studying eating behaviour in what we hope is a naturalistic environment. This facility will include places where we can watch people eating and dining when they don't know we're watching them. And also we can manipulate the environment. For example, manipulate things that they see, things that they smell, the temperature and other factors. We're also doing some more, if you like, clearly experimental studies of behaviour, looking at how the brain responds to certain stimuli. In one recent experiment, we've been giving people high-fat versus low-fat milkshake when they're lying in the scanner and looking at parts of the brain that are activated and seeing clear differences not only between high and low fat, but also between lean and obese people. And finally, really, all of our work is directed towards ultimately helping patients. And to ultimately help patients, we need to understand disease mechanisms, but also to push that knowledge through so we can try and develop therapies. And we're very excited about the fact that after 15 years since we first discovered the first MC4R mutations, there's now a potential compound where we're working in collaboration with Rhythm Pharmaceuticals and also with my chairman here to follow up on these studies done in primates, as shown in this slide. And we hope later this year we'll be able to run a phase 2 clinical trial of a melanocortin receptor agonist to be used in patients with MC4R mutations. So I'm just going to really finish by saying I hope I've shown you that we and others have identified rare, highly penetrant genetic variants in multiple genes associated with severe childhood-onset obesity. We're learning a lot about the mechanisms, and I think we're finding the mechanisms are more complicated than we perhaps first thought, and that the phenotypes in patients are very heterogeneous, and there may be many other things that we're yet to find. But I think what is really key is to try and understand the complexity of human disease. We need to take multiple different approaches and to integrate that knowledge, try to understand how these basic mechanisms translate into physiological effects. And finally, I just want to finish by thanking a number of people, in particular my team on the left, my colleagues in Cambridge, in particular at the Sanger Institute and in behavioural neuroscience, our many collaborators around the world, and in particular the physicians and colleagues around the world who tell us about their patients, without whom we could not do this work. Thank you very much.